Welcome to Essential Mechanisms of Biochemistry. In the previous video, we saw that carbohydrates serve two very different purposes. During the Calvin cycle, ATP and NADPH were invested to reduce carbon dioxide into carbohydrate. In the oxidative pentose phosphate pathway, glucose 6-phosphate was actually oxidized to generate NADPH, while producing 5-carbon sugars for nucleotide biosynthesis. Between those two redox branches lay a remarkable network of reversible carbon rearrangements, where a relatively small collection of chemical mechanisms continually reorganized carbohydrate skeletons. Like glycolysis begins from that same network, but its objective is entirely different. Rather than building carbohydrates or generating reducing power for biosynthesis, glycolysis systematically harvests the chemical energy stored within glucose and converts one 6-carbon sugar into 2 molecules of pyruvate while producing ATP and NADH. Glycolysis begins with hexokinase, another ATP-dependent phosphorylation. An active site base deprotonates the C6 alcohol of glucose, producing an alkoxide capable of attacking the gamma phosphate of ATP. ADP departs as glucose 6-phosphate is formed. Although ATP is consumed, this phosphorylation accomplishes two essential goals. First, it traps glucose inside the cell by introducing a permanent negative charge. Second, it marks the sugar as a metabolic intermediate recognized by downstream enzymes. The investment phase has begun. But before another phosphate can be introduced, the carbonyl must be relocated. As we saw in the pentose phosphate pathway, phosphoglucose isomerase performs an enediol-mediated isomerization. Ring opening exposes the carbonyl. An enzyme base generates an enediol intermediate. Reprotonation shifts the carbonyl from C1 to C2 before the sugar cyclicizes again. Nothing has been oxidized. No carbon has been gained or lost. Only the functional group has moved. That relocation prepares the molecule for the second ATP-dependent phosphorylation. But with phosphofructokinase 1, we have the committed step of glycolysis. Once again, ATP serves as an activated phosphoryl donor. The C1 hydroxyl attacks the gamma phosphate of ATP, producing fructose 1,6 bisphosphate. Unlike the previous reaction, this phosphorylation strongly commits carbon toward glycolysis. Removing the phosphate would require an entirely different enzyme, a bisphosphatase operating during glyconeogenesis. At this point, the investment phase is entirely complete. And now the 6-carbon sugar is divided into two 3-carbon sugars using aldolase. We encountered this exact mechanism during the rearrangement section of the pentose phosphate pathway. A lysine residue forms a Schiff base with fructose 1,6 bisphosphate. After proton transfers generate an enamine, cleavage occurs between C3 and C4. The products are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. One 6-carbon molecule has become two 3-carbon molecules. From this point onward, every reaction occurs twice for each glucose entering glycolysis. Only glyceraldehyde 3-phosphate continues directly through glycolysis. Triose phosphate isomerase therefore rapidly converts dihydroxyacetone phosphate into a second molecule of glyceraldehyde 3-phosphate. Once again, an enediol intermediate shifts the carbonyl without altering oxidation state or carbon skeleton. The investment phase is now complete. We have two identical 3-carbon substrates that continue on into the payoff phase. And now with glyceraldehyde 3-phosphate dehydrogenase, we have the first oxidation of glycolysis. An active site cysteine attacks the aldehyde, forming a covalent thiohemiacetal. A hydride transfer reduces NAD+ to NADH. The resulting thioester is then attacked by inorganic phosphate and produces 1,3-bisphosphoglycerate. Notice what has happened. The energy released by oxidation has not been lost as heat. Instead, it has been conserved within a high-energy acyl phosphate. And now, with phosphoglycerate kinase, the activated phosphate installed during the previous reaction now performs useful work. ADP attacks the acyl phosphate, and ATP is produced. The product becomes 3-phosphoglycerate. Note, this is the first molecule, this same molecule that we made in the first step of the Calvin cycle. This is the first example of substrate-level phosphorylation. Rather than using a proton gradient, ATP is synthesized directly from a high-energy chemical intermediate. But before dehydration can occur, the phosphate must first be repositioned. Phosphoglycerate mutase transfers the phosphate from C3 to C2. The carbon skeleton remains unchanged. Only the location of the phosphate changes. Once again, metabolism prepares the molecule for the chemistry that follows. And now, water is eliminated using enolase. An enzyme base removes the proton adjacent to the phosphate. Hydroxide leaves as water. A carbon-carbon double bond forms, and the product is phosphoenol pyruvate. This dehydration creates one of the highest energy phosphate donors found in metabolism. With pyruvate kinase, the final ATP producing step now occurs. ADP attacks the phosphate of phosphoenol pyruvate, and ATP is formed. The remaining enol immediately tautomerizes to the much more stable keto form of pyruvate. That spontaneous tautomerization provides much of the thermodynamic driving force behind this reaction. If we zoom out, glycolysis is not 10 unrelated reactions. It is a sequence of reoccurring mechanistic themes. We have 2 phosphoryl transfers, 2 enediol mediated isomerizations, 1 Schiff base cleavage, 1 oxidation, 2 substrate level phosphorylations, 1 phosphate migration, and 1 elimination. Collectively, these transformations convert glucose into pyruvate while capturing energy as ATP and NADH. More importantly, though, glycolysis demonstrates the core reaction strategies used throughout metabolism. And once you understand these mechanisms, much of biochemistry becomes recognizable variations under the same underlying chemistry. Till next time, Crip.